Leo myoma. We are Leo myomas. Oh, this has nothing to do with the topic. It's just how I loosen up my vocal cords for the video. So this video is divided into two parts. This is going to be part one. It's just going to discuss hemochromatosis and collagen synthesis. It's probably going to be around 20 minutes long. And there's also a part two of Luffy's medical analysis / plan. It's 8 minutes long. If you don't want the background knowledge of hemochromatosis and collagen synthesis, just watch part two of this video of Luffy's medical analyses. So let's begin. So I'll be discussing Luffy's hemochromatosis. Usually iron is detected with a Prussian blue stain, and for this stain on this patient we may want a smaller tissue because his tissue may tend to expand. So before I talk about hemochromatosis, I want to discuss the properties of iron. So this will be the lumen, this will be the duodenum, and this will be the blood vessel. So the duodenum has microvilli right here, and in order to be absorbed, the Fe3 iron must be converted to Fe2. And one thing that speeds this up is vitamin C. Fe2 gets absorbed by the DMT1 receptor at the surface. And when it gets absorbed, it gets into the cell, it goes through many reactions and turns back into Fe3. Sometimes iron can be stored within the cell, and this usually occurs during infections, so the bacteria can't consume it and grow. This is an evolutionary mechanism our body has adapted, but it's also the mechanism of action for anemia of chronic disease. So let's say you have rheumatoid arthritis, you can get excessive iron storage, so it'll be depleted in the rest of the body, but it'll be excess in the cell, and you don't really treat the iron problem, you treat the underlying cause of the anemia of chronic disease, which would be rheumatoid arthritis, let's say. And in order for iron to leave the cell, it has to go through another channel called ferroportin, which in Latin just means iron gate. And then it gets into the blood vessel. You might be wondering, how does it go into the blood vessel and not damage it? Well, it's usually bound to something like a carrier molecule, such as like transferrin or like ceruloplasmin or hephaestion, and that converts the iron from Fe3 to Fe2, so it can be safely transported. In the liver, there's an acute phase reactant called hepcidin. Anything with the prefix of hep is usually derived from the liver. And hepcidin acts on the ferroportin channels. It causes the ferroportin channels to dissociate from the membrane and go back into the cell, like right here. And it would in the cell it disintegrates. And one mechanism of hemochromatosis is thought to be the HFE gene, which causes a disruption in hepcidin, which would cause excessive ferroportin channels and a lot of iron to go through that channel and would cause hemochromatosis. There would be too much iron compared to carry, and it would just leak out into the blood. So as I discussed, Fe3 gets converted to Fe2. Fe2 is called ferrous. The way I remember this is US as the two of us, and us is also 2 letters long, and Fe3 is ferric, which is the free radical that usually damages the organs and blood vessels and whatnot. The way I remember this is I remember ferricky, and then another song that comes to mind is Icky Vicky, Vicky, Icky Vicky. And so Fe3 gets turned to Fe2, and then it goes through the DMT1 receptor and is catalyzed by vitamin C, and then goes through ferroportin into the blood. and it's carried by ceruloplasmin or hephaestion. And then hepcidin's function is to decrease iron release from the ferroport by disrupting the ferroportin channels. Heme is more readily absorbed from meat than it is from vegetable iron because there's channels. Hemoglobin has two components, heme and globin. Globin is made up of amino acids, and those usually get reabsorbed. And heme is made up of protoporphyrin and iron. Protoporphyrin also gets reabsorbed, and we just discussed what happens to iron. If any of these components are low, you would get microcytic anemia. Iron is usually given in supplements to people with iron deficiency anemia and any other cause like bleeding or whatnot. And usually you give it with a stool softener to prevent the constipation. So now I'm going to talk about hemochromatosis. Hemochromatosis is when iron damages organs or deposits and causes symptoms, whereas hemosidrosis is just it depositing in the tissue and no symptoms, no damage. And iron damages the membrane of the cells. Mutations usually are due to excessive intestinal absorption of iron. And the classic signs are bronze skin and diabetes and cirrhosis. You can also have dilated cardiomyopathy. This is classic hemochromatosis, but it's more commonly associated with restrictive cardiomyopathy. You can also have congenital testicular atrophy. This is usually due to liver damage. So since the liver is damaged, estrogen can't be degraded, and so it builds up compared to testosterone. And you can also get other symptoms as well of liver failure, which is why it's associated with hepatocellular carcinoma. It's an autosomal recessive disorder thought to be associated with the HFE gene on chromosome 6. You can also get hemochromatosis secondary to transfusion, like for example in thalassemia or in like sickle cell. That's why you usually give deferoxamine just so you can prevent the iron over toxicity. And so other organisms to watch out for that are siderophilic microorganisms if you are in a state of hemochromatosis is Vibrio vulnificus, which can cause necrotizing fasciitis, bullous lesions, septic shock, Hep B and C. Males are more prone to hemochromatosis just because females lose blood through menstruation, which has iron in it. That's why you'll see symptoms much earlier in males than in females. And the severity of periodontal disease associated with high transferrin saturation in hemochromatosis patients. So another genetic component is the C282Y allele is a transition point mutation from guanine to adenine at nucleotide 845 in HFE, resulting in a missense mutation that replaces cysteine residue at the position 282 with tyrosine amino acid. So a transition mutation is when a purine turns into another purine. A transversion mutation is when purine turns into a pyrimidine or a pyrimidine turns into a purine. Like if guanine or adenine were to turn into cytosine, uracil, or thymidine. So the lab values for hemochromatosis, you would see high iron, high serum ferritin, high transferrin iron saturation percentage. This just means all the carrier molecules are full, they're carrying iron, and the total iron binding capacity, everything is full. So like there'd be a low total iron binding capacity. So since everything is full, there's not really a lot to bind to anymore. Transferrin would be low. Usually liver makes transferrin in order to, as a scavenger molecule, to recruit iron. And since iron is everywhere, the liver doesn't have a need to recruit it and find it in like an obsolete places. So that's why the liver is going to decrease the production of transferrin. So common symptomology is like with hemochromatosis is like other cirrhotic diseases such as like Wilson's disease, chronic manganese poisoning, and hyperuricemic syndrome in Dalmatians. They can also experience bronzing of the skin. So manganese poisoning, you can see it in welders. It's also a byproduct when making cocaine. and sometimes you can see it in areas with poor water filters. So to diagnose it, you can do a serum transferrin and transferrin saturation in lab work. If you're still unsure, you can biopsy the liver, but usually you don't have to. You can just stop at the serum transferrin and transferrin saturation levels. You may want to get a brain MRI or CT of the bone, just because it may be very deadly if it goes into the brain, and CT bones of the bones. And it's remember that pseudo gout is associated with hemochromatosis, joint trauma, and high parathyroid hormone. So due to it being associated with dilated cardiomyopathy and restrictive cardiomyopathy, you may want to get a echo. Screening for hemochromatosis is usually not necessary unless they have like a first degree relative, such as a parent, child, or sibling with the disease. Treatment would be to get phlebotomy or a phlebotomies. Diet would consist of limiting intake of alcoholic beverages. This is just due to like the liver is already in a poor condition. You don't want to make it worse with alcohol on top. Vitamin C, as we discussed earlier, would cause increased iron absorption in the gut. Red meat contains protein, which contains iron. Same with shellfish and seafood. Shellfish and seafood is also increase the risk of Vibrio vulnificus. So now I'm going to talk about collagen synthesis. So this is the rough endoplasmic reticulum. Let's draw some ribosomes in there. They say no two ribosomes are the same. So from the nucleus, the collagen mRNA synthesis occurs, and then when it gets to the endoplasmic reticulum, it becomes the pre-pro collagen, and then later this is usually made up of glycine and then two other nucleotides, usually proline and lysine, and then hydroxylation occurs. And if this does not occur, you can get scurvy, and it also requires vitamin C. And afterwards, glycosylation occurs, and after glycosylation occurs, there's a triple helix formation. If there's problems with the triple helix formation, you might end up with osteogenesis imperfecta. So after the endoplasmic reticulum, the collagen the the pro collagen molecule right here goes to the Golgi apparatus, where it gets further processed. This is when molecules travel from the endoplasmic reticulum to the Golgi. This is called trans transport, and if it goes from the Golgi back to the ER, this is called cis transport. This is cis. So ER to Golgi, this would be done by the COP2, and from the Golgi back to the endoplasmic reticulum, it would be done by the COP1. So afterwards, from the Golgi, it gets moved into the extracellular space. In the extracellular place, the N terminal and the C terminal get cleaved off, and then it becomes tropocollagen from procollagen. So the reason you cleave the N and C terminals is because like there'll be a little bit of kinking at the end, and this kind of makes it, think of it like as it would be difficult to connect. So like if you have like these smooth connections, your collagen can and link it'll be easier for collagen to link together. So formation of cross links between collagen is stabilized through an enzyme called lysyl oxidase, and this is important for a disease called Menkes disease. Problems with cross-linking can result in Ehlers-Danlos and so the reason we eat keloids with glucocorticoids is because it inhibits collagen synthesis. Elastin is different from collagen in terms that it has interchain crosslinks of lysine, and it has a higher number of non-polar amino acids. And decrease in collagen fibril production results in wrinkling, which is also considered photoaging. So now I'm going to talk about the different type of collagens. There's type 1, which is found in bone. The mnemonic is the word 1 is in bone. Cartilage is type 2 collagen, and then you remember it by saying cartilage. And base has 4 letters, so type 4 collagen is found in the basement membrane. And Marfan and Ehlers-Danlos are both autosomal dominant disorders. Most structural disorders are autosomal dominant, so Marfan is found on chromosome 15. Marfan disorder is a disorder of chromosome 15, and this is usually, you'll see like a tall person with long arms, long fingers. And Ehlers-Danlos, you'll find them to have stretchy like fingers, and they'll be very flexible. There's 2 types, type 5 and type 3. The way I remember type 5 is I imagine like all 5 fingers being stretched out really far, and the way I remember 3 is I put like I draw 2 V's sideways and like this, and I just call V for vascular, so type 3. Both of these are associated with hypertension and many cardiac disorders such as mitral valve prolapse and aortic dissection. So scurvy is a hydroxylation defect. As we discussed previously, it's associated with swelling and bleeding of the gums. They often get sick, easily bruised and bleeding, poor wound healing, pain and swelling of the joints, hair and tooth loss, small bleeding around the hair follicles. They have corkscrew hair and bleeding under the nails, fatigue, dry, and pale skin. So one thing to look out for is that Marfan is associated with upwards eye dislocation. This upward eye lens dislocation, the way I remember this is you have to look up at a Marfan's patient, and this is in contrary to homocystinuria, which has downwards eye lens dislocation. And Menkes disease is an X-linked recessive disorder caused by mutations in genes coding for the copper transport protein ATP7A. Or you want to differentiate this from Wilson's disease, which is a defect in ATP7B and would have excess copper, versus Menkes, which has decreased copper. Characteristics of Menkes include kinky hair, growth failure, and nervous system deterioration, weakened bones, which can cause osteoporosis and fractures. One of the enzymes is lysyl oxidase, which requires copper for proper function. This enzyme cross-links tropocollagen into strong collagen fibers. The defective collagen contributes to connective tissue manifestations of this disease. And that is the end of this video. So watch part 2 if you want to see the analysis of Luffy.